The spinal cord provides a two-way conduction pathway to and from the brain. It is a major reflex center in that spinal reflexes are initiated and completed at the level of the spinal cord. In this lecture series, we will examine the gross and cross-sectional anatomy of the spinal cord, its protective coverings, and the two-way conduction pathways of ascending and descending tracts. We will also look at the origin of the spinal nerves, although the nerves themselves are part of the peripheral nervous system (PNS). Recall that the nervous system as a whole is divided into two major parts, the central nervous system, which includes the brain and spinal cord, and the peripheral nervous system that includes every other bit of nervous tissue outside of the CNS. Use this slide to review the arrangement of the nervous system, and the relationship between the CNS and PNS. The spinal cord in adults is about 17 to 18 inches long, 42 centimeters, and about 3/4 of an inch, 1.8 centimeters in diameter. The spinal cord extends from the foramen magnum of the skull to the level of the first or second lumbar vertebra L1, L2. It does not extend the entire length of because the spinal cord stops growing before the skeleton does. The spinal cord tapers from superior to inferior, finally ending as a cone-shaped structure called the conus medullaris. Nerve roots arise from the spinal cord and exit the vertebral canal through the intervertebral foramina at the level of the body that the nerves control. In other words, each spinal nerve exiting the vertebral canal innervates its corresponding body somite segment. During fetal development, however, the vertebral column grows faster than the spinal cord, so the lower spinal nerve roots have to travel farther and farther inferiorly through the canal to reach their exit points in order to get to the body somite that they control. The collection of nerve roots found in the inferior end of the vertebral canal is called the cauda equina, which literally means horse's tail. Another structure visible arising from the conus medullaris is the filum terminale. It is a fibrous extension of the pia mater that anchors the spinal cord to the coccyx, preventing the cord from being displaced by body movements. Both the brain and the spinal cord are protected by bone, cerebrospinal fluid, and special CT coverings called the meninges, singular is meninx. The spinal cord and its meninges lie in the vertebral canal. Unlike the brain's meninges, the dura mater of the spinal cord is a single layer and is not attached to the bony walls of the vertebral column. However, it is continuous with the dura mater covering the brain. The spinal dura mater extends to sacral level 2S2. In the sacral canal, the spinal dura tapers, forming a dense cord that blends with the filum terminale, forming the coccygeal ligament. The dural connections at the foramen magnum and the coccygeal ligament prevent longitudinal superior-inferior movement of the spinal cord. The middle covering is the arachnoid mater. The inner surface of dura and outer surface of arachnoid mater are covered by simple squamous epithelium, and between them is the subdural space. Recall from our discussion of the brain meninges, that the arachnoid mater, which means spider mother, gets its name from the loose CT and elastic fibers called arachnoid trabeculae, that resemble a spider's web. The arachnoid trabeculae extend between the arachnoid membrane and the outer surface of the pia mater. Between the arachnoid and pia is the subarachnoid space, which is filled with CSF. As with the brain, cerebrospinal fluid CSF circulates through the subarachnoid space, as well as internally through the central canal of the spinal cord. Consequently, both the brain and the spinal cord are floating in CSF. Besides acting as a shock absorber, CSF helps nourish the spinal cord. It is a medium for the diffusion of oxygen and carbon dioxide gases, nutrients, chemical messengers, and it washes away waste products produced by neuronal metabolism. This fluid can also be used for diagnosing various diseases, such as infection, bleeding, electrolyte imbalance, etc. CSF can be safely removed from the subarachnoid space by a procedure called a spinal or lumbar tap or puncture. Because the spinal cord tapers inferiorly, finally ending at L1, L2, a needle can be inserted in the region of the cauda equina without the risk of puncturing the spinal cord. The patient being tested assumes the fetal position by bringing their knees as close to their trunk as possible. A needle is inserted and CSF is removed for testing. Just like the pia mater found covering the brain, the spinal pia is thin, transparent, and adheres tightly to the surface of the spinal cord. It contains branches of the blood vessels that supply the spinal cord. The spinal cord is secured to the dura mater throughout its length by sawtooth slips of pia mater called denticulate ligaments. Denticulate means toothed. The denticulate ligaments prevent lateral or side-to-side movement of the spinal cord, preventing it from smacking into the hard bony surface of the vertebral canal when we move. Let's take a look at what the spinal cord looks like on the inside. This picture shows cross-sections of the spinal cord at different levels. The spinal cord is mostly cylindrical in cross-section except for enlargements in the cervical and lumbar regions. These enlargements contain neurons that form the mini nerves that control the complex musculature of the upper and lower limbs. The spinal cord has two grooves, the ventral anterior median fissure, and a shallower dorsal posterior median sulcus. These grooves run the length of the spinal and almost divide it into right and left halves. Locate these features on the diagram above. Internally, the spinal cord contains gray and white matter, the arrangement of which is opposite that of the brain. Recall that in the brain, the gray matter of the cerebral cortex is located superficially, whereas the white matter is deep. In the spinal cord, the gray matter makes an H or butterfly shape and is surrounded on the outside by white matter. Just like in the brain, the gray matter of the spinal cord consists of neuron cell bodies, neuroglia, unmyelinated axons and the dendrites of association fibers. The white matter consists of myelinated axons of sensory and motor neurons. The two large lateral masses of gray matter are connected by a bridge of gray matter surrounding the small central canal located in the center. These crossbars of gray matter are called the gray commissures and they contain axons that cross from one side of the cord to the other on each side of the central canal. The projections of gray matter are called horns. The ventral horn contains somatic motor nuclei cell bodies of motor neurons. They form columns of gray matter that run the entire length of the spinal cord. The amount of ventral gray matter present at a given level of the spinal cord reflects the amount of skeletal muscle being innervated at that level. The posterior or dorsal horns extend through the white matter to the surface of the spinal cord. They consist mainly of interneurons that relay somatic and visceral sensory information. Afferent fibers carrying impulses from peripheral sensory receptors form the dorsal roots of the spinal cord. The actual cell bodies of the sensory neurons are found in an enlarged region called the dorsal root or spinal ganglion, which lies outside the spinal cord and is thus part of the peripheral nervous system. After entering the cord, their axons travel in several white matter tracks to synapse at higher levels or with interneurons at the same level. In the thoracic and superior lumbar segments of the cord, there is a lateral horn of gray matter. The lateral horn neurons are autonomic, sympathetic motor neurons supplying the viscera, smooth muscle or glands. Their axons leave the cord via the ventral root along with those of the somatic motor neurons. The dorsal and ventral roots are short and fused together to form a mixed spinal nerve. The gray matter neurons comprise nuclear clusters of neuron cell bodies with similar functions that are organized based on their function in the part of the body they are innervating. Thus, the gray within each horn is divided into four zones: somatic sensory SS, visceral autonomic sensory versus, visceral motor VM, and somatic motor SM. Because of this high degree of organization of nuclei and fiber tracks, it is easy to predict which areas of the body and what functions would be affected by damage to a specific area of gray matter located in a specific level of the cord. The gray matter of the spinal cord is divided into a sensory half dorsally, and a motor half ventrally. This diagram shows clusters of neuron cell bodies organized into functional regions seen on the right side, and the organization of their axons within the spinal roots and spinal nerves on the left. Remember that the dorsal and ventral roots, as well as the spinal nerves, are part of the peripheral nervous system (PNS), not the CNS spinal cord. Let's examine the dorsal horn on the right. SS stands for somatic sensory, and this region contains interneurons that receive sensory input from somatic neurons. Next, is the visceral sensory region. It too consists of interneurons, but these receive sensory input from the organs. Notice that the cell bodies of both somatic and visceral sensory neurons form the dorsal root ganglia. Recall that sensory neurons are unipolar neurons and have their cell bodies off to the side of the axon. In the ventral horn, the cell bodies of visceral motor or autonomic neurons are shown in yellow. These neurons regulate activities occurring in the organs. The next part of the ventral horn contains the large cell bodies of somatic motor neurons. These neurons control voluntary skeletal muscle contraction. Now let's take a look at the organization of the white matter of the spinal cord. As seen in the brain, the white matter of the spinal cord is composed mainly of myelinated axons that carry information back and forth between different parts of the spinal cord and the brain. The fibers run in three directions. One, ascending tracks carry sensory input from peripheral receptors to higher centers of the spinal cord and brain. 2, descending tracks carry motor commands down to the cord from the brain or within the cord to a lower level. 3, tracks that run transversely cross from one side of the cord to the other. Like we saw in the brain, fibers that cross the midline to connect one side to the other are commissural fibers. Recall the corpus callosum that connects the right and left cerebral hemispheres. The fibers are grouped into 3 white matter columns on the right and left side of the cord, also known as funiculi, equals long ropes. Each funiculus is named according to its position I. In the cord, dorsal posterior, lateral and ventral anterior. All of the major spinal tracts are part of multi-neuron pathways that connect the brain to the body's periphery. Most fiber tracts or pathways cross from one side of the cord to the other at some point along their journey. This crossing to the contralateral side is called a decussation. Remember the decussation of the pyramids when we studied the brain. In addition, most pathways exhibit somatotopy, which is a precise spatial relationship among the tract fibers that reflects the orderly mapping of the body analogous to the homunculus of the cerebral cortex. Finally, all pathways and tracks are paired and symmetrically arranged in the right and left sides of the spinal cord. In this diagram of a transverse section through the spinal cord, the ascending sensory tracts are colored blue and the descending motor tracts are colored red. Note the bilateral symmetry of both types of tracts. Each column or funiculus is made up of several fiber tracts, each called a fasciculus stick. The fasciculi contain bundles of axons with similar destinations and functions. Another thing to note is the naming convention of the tracts. They are named based on their relative position in the spinal cord, dorsal or posterior, lateral, and ventral or anterior, and where they are coming from and going to. For example, the posterior spinocerebellar tract indicates that this bundle of fibers is an ascending tract spino, carrying sensory information brought into the spinal cord to the cerebellum for processing. Similarly, one can surmise that the lateral corticospinal tract is a descending tract because it is coming from the cerebral cortex and going to the spinal cord. You know it is carrying voluntary motor commands because descending tracts are motor. Remembering this naming convention will help you determine the general function and the kinds of information the tracts are carrying. We will consider ascending or afferent pathways first. Ascending pathways conduct sensory impulses upward, typically through chains of 3 neurons designated first, second and third order neurons. Both second and third order neurons are interneurons. First order neurons have their cell bodies in dorsal root ganglia or cranial nuclei and conduct impulses from cutaneous receptors and proprioceptors to the spinal cord or brainstem, where they synapse with second order neurons. The cell bodies of second order neurons reside in the dorsal horn of spinal cord or in nuclei located in the medulla oblongata. They transmit impulses to the third order neurons in the thalamus that relays the impulses to the somatosensory cortex. In general, somatosensory information is conveyed via 3 main pathways: the spinocerebellar, medial lemniscal and spinothalamic pathways. Notice that 2 of the pathways, medial lemniscal and spinothalamic, carry information that is relayed in the thalamus to the somatosensory cortex of the brain. These pathways carry sensory information that we become consciously aware of. Now look at the spinocerebellar pathway indicated on the left side of diagram A by the light blue line. This pathway never reaches the thalamus or somatosensory cortex of the brain. The spinocerebellar pathway transmits proprioceptive muscle or tendon stretch information only to the cerebellum, so it is subconscious. The cerebellum uses the information to coordinate skeletal muscle activity. Examine the right half of diagram A. This is the dorsal column medial lemniscal pathway. The term lemniscal comes from a word that means ribbon. It transmits discriminative touch and vibration, as well as conscious proprioception position sense to the cerebral cortex, which allows us to determine precisely where on the body the sensation originated from. This pathway is a little more complex. Follow this pathway, which is indicated by the dark blue line. The dorsal column medial lemniscal pathway is divided into 2 tracts that are named for the nuclei in the medulla oblongata, where the first order neuron synapses with the second order neuron. The fasciculus cuneatus is a shorter pathway because its first order neurons carry sensory information from the upper limbs, upper trunk and neck. The first order neurons synapse in the nucleus cuneatus of the medulla. From the medulla, The second-order neurons of both tracts decussate cross over and continue their ascent to the thalamus in the medial lemniscal tract. Once in the thalamus, the second-order neurons synapse with the third-order neurons which ascend to the appropriate region of the somatosensory cortex post-central gyrus. Figure Re-B in the diagram shows the spinothalamic tract. This lateral pathway carries pain and temperature information into the dorsal horn of the spinal cord where the first-order neuron synapses with second-order neurons. The fibers of the second-order neurons decussate cross over to the other side before ascending to the thalamus where they synapse with the third-order neurons. These carry the information to the appropriate region of the somatosensorial cortex. This figure shows three major descending pathways by which the brain regulates movement of the body. Descending pathways differ from ascending pathways in that they use only two neurons to transmit information instead of three. In these two neuron pathways, the neurons are referred to as the upper and lower motor neurons. The pyramidal cells of the motor cortex as well as the neurons in subcortical motor nuclei, basal nuclei, etc. are called upper motor neurons. The ventral horn motor neurons which innervate the skeletal muscles, their effectors, are called lower motor neurons. The direct or corticospinal pathways are also called the pyramidal tracts because the upper motor neurons are the large pyramidal cells of the somatomotor cortex pre-central gyrus. The axons of the pyramidal neurons comprising the lateral corticospinal tract decussate in the pyramids on the ventral surface of the medulla oblongata. From there, they descend to the appropriate level of the spinal cord. They synapse either with short ventral horn interneurons, or directly with the large multipolar neurons of the ventral horn which send impulses out the spinal nerve to skeletal muscle. Because of the decussation of the pyramids, the right cerebral cortex controls skeletal muscles on the left side of the body and vice versa. Unlike the lateral tracts, the anterior corticospinal tracts do not decussate in the medulla, but cross over at the spinal cord level to synapse directly with the ventral horn motor neuron. It is clinically important to know where the fiber tracts cross, as we shall see later. The other motor tract shown in diagram B, is the rubrospinal tract. This tract is an example of an extrapyramidal or indirect pathway, so named because the origin of their neuronal nuclei were thought to be outside of the pyramidal tracts. Recent evidence suggests that this may not be entirely accurate, but the term is still widely used in clinical settings. In general, the indirect pathways are involved in regulating the axial muscles more or less automatically. For example, the vestibulospinal tract carries information that helps us to maintain balance and posture, the reticulospinal tract regulates the muscles controlling coarse limb movements, and the tectospinal tract regulates head, neck, and eye movements that follow objects in the visual field. Many of the activities controlled by subcortical motor nuclei are involved with reflex activity. In the rubrospinal tract, the origin of the upper motor neuron is in the red nucleus of the midbrain. Their fibers decussate just inferior to the red nucleus. From there, they continue through the brain and down the spinal cord. Upon reaching the correct level, the fibers synapse on a short interneuron which synapses with the ventral motor cells. The rubrospinal tracts are concerned with muscle tone and flexors on the opposite side of the body. Although the spinal cord is elastic and stretches when we bend or move our trunk, it is delicate and sensitive to pressure. Damage to the spinal cord or its roots can lead to some sort of functional loss. Paresis and paralysis are varying degrees of motor function impairment. Paresis pertains to muscle weakness, whereas paralysis is more severe. Paresthesias is loss of sensation. There are different types of paralysis. If the cell bodies of the ventral horn or the ventral root are severely damaged, then the patient develops flaccid paralysis of the muscles that were being served at the level of injury. Flaccid paralysis results in a loss of voluntary control of the affected skeletal muscles, no reflex activity, the muscles remain limp and will eventually atrophy. This condition is also known as lower motor syndrome. If, however, only the upper motor neurons coming from the primary motor cortex are damaged, the individual develops spastic paralysis. What happens is that the motor neurons remain intact and the muscles continue to be stimulated. However, the stimulation is irregular and non-voluntary because it lacks central control. The muscles remain healthy longer, but their movements are spastic. Frequently, this leads to a permanent shortening of the muscles. Transection of the spinal cord at any level results in total motor and sensory loss in the body regions inferior to the site of damage. If the transection occurs between T1 and L1, both lower limbs are affected, resulting in paraplegia. If the injury occurs in the cervical region, all four limbs are affected and the result is quadriplegia. Hemiplegia is paralysis of one side of the body. This is usually the result of brain injury like from a stroke.